A very warm good morning to all the participants of this one-day international webinar. Well, we all know that humanity needs leadership, but solidarity is needful to defeat coronavirus. The COVID-19 pandemic is defining global health crisis and is being looked as the greatest challenge of our time. Except for Antarctica, every continent is reeling under this unprecedented crisis. The current statistics state that there are around 17 million confirmed cases in the whole world, with around 6 lakh 67,000 fatalities. While looking at the statistics in India, there are 1.58 million confirmed cases with nearly 35,000 fatalities. It was in December 2019 that the Chinese authorities notified the world that a virus was spreading in their communities. In the following months, it spread to other countries. This virus is the severe acute respiratory, also known as SARS coronavirus 2, simply called coronavirus, causing COVID-19. Its high infectivity rate and impact caused increase in caseload all over the world. This property caused coronavirus to be noticed internationally. It was on March 11, 2020, the World Health Organization declared it a pandemic. The mortality rate is estimated to be around 1 to 6%. All these are surely raising questions about community spread. The worries that public are being subjected to community transmission is creating a fear. The thin line between caution and panic needs to be exposed and addressed. We all are very well aware that COVID-19 is contagious, but just how contagious is COVID-19 is a big question. Looking into the rise in asymptomatic cases, how this contagion affects our immune system, are there any vaccines? Will they really help? Well, these are some questions which we would like to know. So today we are fortunate to have three distinguished speakers on our platform and also more than 1,200 registered participants who, staying safe at their homes, are anxiously waiting to hear the speakers. So to kickstart off with this webinar, I first request our principal, sir, Dr. M.G. Royman, to give a formal welcome to our esteemed guests and participants. Sir, please.
A warm good morning to all the speakers and participants taking part in this international webinar organized by PG Department of Microbiology and Biotechnology on the topic Epidemiological and Immunological aspects of COVID-19. At this point, I would like to mention that St. Thomas College, Bhilai was established in the year 1984 and has emerged as an institution of excellence in the field of higher education since then. The college has been awarded B++ through re-accreditation in 2017. St. Thomas College is proud of being awarded the Best College Award in the year 2014 amongst the several private colleges affiliated to Pandit Ravi Shankar Shukla University, Raipur. This self-financing educational institute has always shown its excellence in academics by achieving merits at university level. I take the opportunity to welcome all the eminent speakers of today's international webinar. I would like to welcome our first speaker, Dr. Manjula, Associate Editor at Bioprotocol, New York. I also welcome our second speaker, Dr. Harsha Ramchandani, a dedicated Chief Medical Officer at Bay Area Community Health, California. I also take the opportunity to welcome our third speaker of today's webinar, Dr. Sunil S Porad, associated with Institute of Bioresources and Sustainable Development, Imphal, Manipur. It gives me immense pleasure and honor to introduce all the speakers who will be enlightening our participants with the epidemiological and immunological aspects of COVID-19 and also briefing us with the recent development related to the ongoing pandemic. The blessing of Manager Bishop, His Grace Dr. Joseph Mar Dionysius, are always with us. I welcome your grace to this session. Administrator, a very Reverend George Matthew Ramban, his constant source of inspiration, Father Rambachan, I welcome you. I also welcome all the faculty members, dean, academics, IQC coordinator, research scholars, and students who make up the majority of the participants of today's international webinar. I hope that this webinar will be a great success and purposefulling for all the beneficiaries. Thank you.
Thank you, sir. May I now introduce our participants. May I now introduce our participants with our first speaker, Dr. Manjula Mommadi. Dr. Manjula, who is professionally skilled and protein biochemistry, molecular biology, proteomics, and mass spectrometry, did her MS in molecular sciences and nanotechnology from Louisiana Tech University, Los Angeles. She went on to complete her PhD in biochemistry from Louisiana State University, Los Angeles. Before proceeding for her MS and PhD, she worked as cancer research scientist at University of Texas Health Sciences. Her work concentrated on pediatric bone cancer. She is adept in handling cancer cell lines and building methodologies to study gene therapy. Dr. Manjula also held the post of postdoc scientist and lab manager for 2 years from 2016 to 18 at Rutgers University, New Jersey. Her excellent ability to analyze data helped her to assist in developing SOPs and grant proposals. Currently, Dr. Manjula holds the post of Associate Editor for Bioprotocol, which is an online peer-reviewed journal for protocols, where she has solicited and evaluated more than 50 publications. We are fortunate to have Dr. Manjula as one of the speakers who, with such an impressive background, will be enlightening us with the recent facts of coronavirus. Dr. Manjula, please.
Thank you so much for lovely introduction. Am I sharing the screen? Okay, here it is. So is my voice clear? I just wanted to check.
Yes, madam, your voice is clear.
Okay, perfect. Okay, so I will be talking about SARS-CoV-2 mechanistic insights and its imprint on molecular passports. We will quickly go over some of the terminology. So the virus name that causes COVID-19 is SARS-CoV-2. It causes the coronavirus disease. It's a pandemic. So I wanted to quickly go over the difference between epidemic and pandemic. So an epidemic occurring worldwide in a wider area between different countries is called pandemic. So why is it called coronavirus? It is called coronavirus because of its crown-like appearance that we see in the structure. It is made of a bunch of proteins here, which is spike protein, which are like spikes. It has membrane protein, nucleocapsid. It has RNA as its genetic material, and it's considerably larger virus, like about 100 nanometers in size. But all of these proteins have different functions, and we will go over that in our next slides. Most importantly, the spike protein is of interest because of its binding and fusion properties. So, so coronavirus is not new to us. It is something that is known for a long time. There are many different types of coronaviruses, but some of them are human coronaviruses, like it's shown here. Novel coronaviruses are classified based on its infectivity to humans. One of them is MERS. In 2002 or 2003, actually, SARS-CoV was identified, and there's a lot of research done on it. Another new coronavirus is called novel coronavirus because it came next to SARS-CoV. It's called SARS-CoV-2, which is what we would be talking about today. So I wanted to go over a little bit of details. There is a high similarity between SARS-CoV and SARS-CoV-2. The origin is sort of unknown, but we know that it came from bats. There are bats infected some other species, which is sort of unknown, but eventually it infected humans. So it all started in Wuhan, and there was the seafood market where bats, it was thought that the bats infected some animals, and then after eating seafood or other animals, humans were infected. Infected patients had severe pneumonia. So initially it was thought that maybe only patients who are infected with pneumonia have coronavirus or SARS-CoV-2. But then later on, it was identified patients without pneumonia were also infected with SARS-CoV-2. And that's when it was identified that it's human-to-human transfer of the infection. Now, coronaviruses, which are found in birds but not in bats, are of different classes. Coronaviruses are classified into alpha, beta, gamma, and delta. So the ones coming out of bats are alpha and beta. The ones coming out of other birds are gamma and delta, and these do not infect humans. The SARS-CoV-2 that we are talking about today is beta coronavirus. Okay, so today I would be talking about the entry. It would be mostly molecular aspects of the coronavirus. We would be talking about the entry of the coronavirus, its replication and gene expression, its immune interactions, and a little bit of therapeutics. Okay, so at the molecular level, how does SARS-CoV enter the body and its replication process? So we have seen the structure of coronavirus before. It has spike proteins, and before entering the cell, when it sits on the cell, there are a bunch of receptors which are called ACE2 receptors, or angiotensin converting enzyme. So when the spike protein interacts with this particular receptor, the entire virus is internalized, and then the genetic material is released, which is single-stranded RNA. And the genetic material of the virus uses the machinery from the host to replicate its genome and then produce all the different proteins that we talked about earlier, which is the translation products are then assembled in the Golgi compartment, and then the mature virion is formed, which is eventually released. So basically, virus does not have its own machinery to replicate. It uses host machinery to replicate, and then it forms a mature virus and it releases. Now, once the cell is infected, how are the other cells infected? We will see that soon. Quickly going back, like we saw here that ACE2 receptors are the ones that interact with the spike proteins. It was initially thought that only lungs have these ACE2 receptors, and probably that's why it's infecting just the lungs. But we know from the recent news that it can infect or affect other parts of the body as well. So ACE2 expression was looked at in various parts of the body, and anything colored in the human body shows the expression of ACE2 receptor. Brain does not have ACE2. Heart, liver, intestines, lungs, kidneys, and the vascular system, all of these have ACE2 receptors. And we know now from recent news that all of these parts can be affected. Okay, so there is a lot of information about the similarity between SARS-CoV and SARS-CoV-2. SARS was the infection which happened in 2002. And because of a lot of research done, then we know we have been able to understand a lot about SARS-CoV-2. So I talked about ACE2 receptors, but there is another receptor or another protein that's expressed on the human cell, which is called TMPRSS2. And this is the priming protein that assists the spike protein to interact with ACE2 and then perform the entire process of replication that we saw in the previous slide. So one more thing is recently there were vaccine that was developed against this particular protein in SARS, and it showed very high antibody generation to take care of the SARS-CoV infection. However, when the same vaccine was used for SARS-CoV-2, it only showed moderate infection. When I say vaccine, I meant antibodies. Okay, so activation of coronavirus spike proteins by host cell proteases occur at different stages of the viral cell cycle. So we talked about how a viral cell, a virus infects a cell, but now in the body, once a virus infects a cell, how are the other neighboring cells infected? So there are many cells and proteases and enzymes, and you will see soon there are many factors that are influencing how target cells are affected and how our body responds to the virus. So once we have virus replicated and it gets out of the cell, then it can affect the neighboring cells. So again, the same process. There is the spike protein, which would interact with the ACE2 receptor. And then there are other proteases around which actually interact or cut at a specific site on the spike protein. Spike protein has those certain sites, and furin and TTSPs can interact with those, cut those sites, and that actually activates the spike protein. And that causes changes in once spike interacts with receptor. And again, the same process continues with genome replication, expression of the proteins related to the virus, viral assembly, and then again, it's released from the cell. So we'll go a little bit over the spike protein now that we have been talking a lot about how spike protein interacts with the ACE2 receptor. This is a crystal structure of spike protein from virus. So this is the sequence, the protein sequence, amino acid sequence of the spike protein. It contains a signal peptide, and basically the entire sequence is divided into receptor binding, meaning binding to ACE2, and then fusion. So there are certain sequences. Just give me a minute, please. Are you able to hear me? Hello?
Yes, madam, we are able to hear you.
Okay.
Just full screen your slideshow, madam.
Yeah, I was on full screen slideshow.
But we are not getting the full screen, madam.
So I was on full screen.
You can go to slideshow and then
Okay, on my computer, I'm seeing slideshow. Are you able to see it too?
no, we are seeing the PPT, but not in the full screen, madam. You just go to the second last menu, that is slideshow, and just go full screen, madam, from there.
Okay, slideshow, which is this. Okay, let me try again. Just give me 1 minute, please. Sorry.
Okay,
And
ma'am.
on my end, I'm seeing full screen slideshow, but it's not showing up here. Okay, so okay.
Uh,
Okay, now
you
my
have to
computer
share
is
it
stuck.
again.
I'm
Yeah,
sorry.
you have to share it again, madam, in this broadcast studio.
Yeah. Okay, so I'm splitting my screens and I'll see if that helps. So I have share screen, application window, this share. Okay, I'm going to make it full screen. Do you see it now?
It's not full screen, but you carry on, madam. No problem. Let it be like this. It's visible.
Okay.
You continue, madam.
Okay, I'm sorry about that because on my screen I see full screen, slideshow. Give me like 30 more seconds. Let me try.
You can change the layout, I guess.
So, no. If it's not coming, I might as well get rid of this. Is this visible better if it's not showing?
Yes, madam. Yes, madam. You continue. It's good.
Okay. Okay, I think it's visible.
Yeah.
Okay.
Yes, madam.
Okay, so this is the crystal structure of spike protein. So if we look at the virus has, this is the viral membrane. Let's say you have a virus here, and then this is the spike protein. And let's say you have a human cell here. So it would have ACE2 receptor binding the spike protein here. So the interaction region between the ACE2 receptor from human cell and from the spike protein is called RBD, which is receptor binding domain. So it would be binding receptor, so it's receptor binding domain. Now it is a dimer, like you can see that there are two colors, one is green, one is blue. And then there are also special sites, R667 and R797, which is arginine. These special sites are the regions which are common in all coronaviruses. And this is highly specific, where it can cause, the cut sites that I talked about earlier from proteases. And it is highly conserved. So this is a little bit about the spike protein, its interaction with the ACE receptor. There was a clinical study performed recently, like a couple of months back, where from about 100 patients were tested, and patients who have recovered from COVID-19 infection. So recovering patients were their antibodies were, their plasma was extracted, and their antibodies was looked at. So the region of its binding to the spike protein is at the different epitopes. So the epitope is the region of the antigen where the antibody would bind. So these are 3 different antibodies which are coming from different patients. And all of these 3 antibodies bound to the spike protein very specifically, but then it's binding to the different epitopes of different regions of the spike protein. So what it tells you is, although different patients are generating different types of antibodies, but it is still going against the spike protein. Okay. Okay. So then there was this another study which was comparing SARS-CoV-2 and SARS-CoV, and they wanted to understand the binding affinity of ACE2 receptor with the spike protein. And what they found is, in both SARS-CoV and SARS-CoV-2, there was very high affinity of binding of ACE2 receptor with the spike protein binding region. However, remember I mentioned that there are alpha, beta, gamma, and delta type of coronaviruses. Now these are coming from the gamma and delta coronaviruses. So this is from alpha and beta coronaviruses. Now these are very highly specific to bind to ACE2. However, here you can see that these are not specific. And like I mentioned, that gamma and delta coronaviruses do not infect humans. Now this is the same information providing information on relative binding between different coronaviruses. And it's very clear that there is a high affinity binding to SARS-CoV-2. So similar study, what they did was they took yeast cells, and then they expressed the RBD region, or the receptor binding domain region from the spike, and expressed it on the surface of the yeast. And then they performed in vitro studies. The Star et al. Group performed in vitro studies where ACE2 proteins were provided in the media. So what they tried to study was, once the yeast is expressing the RBD region, so it is a stretch of amino acids. Now what you could do is perform a mutation analysis where you could individually target each amino acid at a time and change it to anything you like. So there are 20 amino acids, you could change it to anything. So the entire RBD region was changed specifically in the mutation analysis to different amino acids. And then they asked the question that whether it could bind to ACE2 receptor or not. So based on this analysis, they found that there are residues which affect the binding affinity to ACE2 receptor, which may or may not affect the immunogenicity. There were also residues which enhanced the affinity to bind RBD and ACE2. So what we can learn from this is you could probably find potential targets, drug targets of these residues. RBD is also the most variable region in coronavirus. So mutation screen performed in the RBD region was very useful. So this is like a open database, public database that anybody could use, and they could look at and design the drug targets. Okay, so then I wanted to get into the immune response. We looked at how spike protein interacts with ACE2. Now, once the interaction happens, how does our body react to it? So whenever there is an infection from bacteria or virus, the first response that happens is innate immune response, in which you would have establishment of the infection first. So if you're looking at innate immune response, if you are taking it from COVID-19 perspective, initially it needs to have like a threshold level of viral particles that would infect the human host cell. And then once it has infected, then there is an establishment of infection, meaning that virus would go and it would replicate. It has to reach a certain level to activate the adaptive immune response. So once adaptive immune response is activated, that's when your antibodies are generated. So there is a certain timeline. Innate immune response can go up to 4 to 8 days, and then adaptive immune response kicks in. Okay, so now that we know these two words, innate immune response and adaptive immune system, so there are different types of cells that are activated in response to any pathogen that infects the cell. There are dendritic cells, macrophage cells, T cells, natural killer cells. And in adaptive immune system, there are T and B cells. There are different types of T cells. Okay, a little bit going into a lot more detail of innate immune response is once the virus interacts with ACE2, enters the cell, there are toll-like receptors which would identify and then activate other bunch of proteins, which are sensors to activate another set of proteins. And then eventually it would activate interferons, which would further activate the JAK-STAT pathway. And then eventually a lot more interferon genes are expressed or stimulated, which work towards killing the virus. Now, this is the initial host response. Now, let's say virus is not killed or it's taking longer, then there is antibody-mediated immunity or humoral immunity that is activated. So once human cell is exposed to COV2 RNA, then you see that there are two types of antibodies that are generated, IgM and IgG. So after about 4 to 5 days, we start seeing the level of IgM increasing. And then later on, IgG starts increasing, and that becomes, and that is the one which would fight against the infection. So during the process, memory B cells should be, ideally, if it was not SARS-CoV-2, memory B cells would retain the information, and in future, if the same infection happens, it would fight against it. But we still do not know anything about in case of SARS-CoV-2. So virus-specific IgM, IgG are detected within 7 and 14 days. Viral RNA is inversely correlated with the neutralizing antibody titers. Higher titers have been observed in critically ill patients. Okay, so now a final summary slide that I wanted to have to show that how many types of cells that are activated in response to the virus. So there are a bunch of different types of T cells, CD4 plus and CD8 plus type of cells, B cells, natural killer cells. So what happens is initially, once you have innate immune response, it would activate the B cells and T cells. Now, there are two types of T, CD4 plus and CD8 T cells, and both of them activate different types of interleukin, tumor necrosis factor, interferons, and all of these can fight against virus. And then there are many other types of cells and so many different types of cytokines that are activated. And when we hear about the word COVID storm, or cytokine storm, this is what happens when the cells are releasing, when the body is releasing so many different types of cytokines, which actually comes from a type of T cells. And then there are a bunch of different antibodies, and we will talk about that in a little bit. So, so now that we talked about different types of cells, there was a study that was performed in about 125 patients. And in this study, only healthy or recovered patients were looked at. When you're looking at these patients, you could extract their blood, look at the type of cells, the type of CD4 plus, or the type of T cells or B cells you could look at using flow cytometry. So basically, a cytometer is something that could divide cells, separate cells based on color, based on, for example, if a cell is expressing fluorescence, then it would separate out the cells based on red or green fluorescence, whatever fluorescence it's generating, or the size of the cells, and many other factors. So they used cytometry and separated the samples based on CD4 and CD8 expressing cells. So a lot of patients were expressing both B and T cells. They had, expressing is the wrong word, but they had activated or more numbers of B and T cells. Some of them had only B but not T. Some of them did not have both B and T. And this is how they were able to categorize whether their body is able to fight or how it is able to fight the COVID infection. Okay, so next I wanted to talk about, how SARS-CoV-2 is a relatively new infection that we see, and we know of it only since December, mostly since past 6 months. And it has, in fact, it has moved on to different countries in probably like last 3 or 4 months. And that's when all of this research started. But how is it that we know a lot more about SARS-CoV-2 in like 3 or 4 months? That is because we know a lot of information from SARS before, which happened in 2003. It was an epidemic. There were about, the rate of SARS infection was like 9% of the population. There were probably, there were a few countries that were affected, but in case of SARS-CoV-2, there are like more than 100 countries affected. So I wanted to go a little bit over how there is a relation between SARS and SARS-CoV-2 at the sequence level. So this is a protein sequence of the RBD region of the spike protein. Now we can see MERS-CoV spike protein sequence. We can see SARS-CoV, we can see SARS-CoV-2. All the cyan color or the blue color that you see are the regions which are similar but not the same. So for example, when I say similar, you have similar type of residues. For example, if I say acidic residues, so it would be glutamate and aspartate. So, and you could see aspartate in one case, glutamate in another case. Similarly, for example, if you're looking at basic residues, then arginine, lysine, something like that. So they are similar based on its properties. And then, so there are a lot of sequences which are same. And then the star ones are highly conserved. Cyan ones are similar. And so the SARS-CoV-2 and SARS-CoV has more than 70% sequence similarity, meaning that it is also structurally similar, meaning that if you have an antibody for SARS, it would potentially work for SARS-CoV-2 unless there are point mutations or a mutation that would cause major structural changes. MERS-CoV is not that similar to other SARS-CoV viruses. So there was this experiment using Western blot, where you can see that how antibodies react with, just a minute. Okay, how antibodies react, antibodies against SARS-CoV would react with SARS-CoV-2. So this is just a gel where proteins, where the vectors expressing MERS-CoV RBD region or SARS-CoV RBD region or SARS-CoV-2 RBD region is expressed. And when you add antibody against SARS-CoV only, this band is highlighted for both SARS-CoV-2 and SARS-CoV. So like I mentioned, that if there is a structural similarity, antibodies who function based on pattern recognition of the structure. So the antibody for this was used for all three of these, and what we see is the band highlighting for both of them. However, when we had antibody for MERS, it only highlighted for MERS, and we do not see its interaction with other two. So what it can tell is we could use information from SARS-CoV, any drugs that worked for them or vaccines that worked for them could be the initial steps to treat SARS-CoV-2. Okay, so there are many different types of vaccines like viral vectors, where you could express the entire spike protein and express the antigen in the host, and that's how viral vectors work, or genetic vaccines where you could have mRNA, which is what I would be giving an example in a later slide, and then recombinant protein, or attenuated or inactivated or viral-like particles. So there are many different types of vaccines, but we would be talking about a couple of them. The most successful one, I would say, so far in the next few slides. So there is this recent study, actually it was published today, but the clinical trial study was published like 10 days back. So this is from a replication deficient chimpanzee adenovirus, which expresses spike protein, and that vaccine was injected in 1,000 individuals with no sign of COVID-19. So then what happened, even after the single dose of this vaccine, it elicited response against the spike protein. So there were spike-specific antibodies in many individuals. And the individuals, okay, so here it says seen in 91% of the individuals. And after the second booster dose, all of the individuals showed high titer of antibody. Now this would have to be tested. This is a, I'm sorry. So this would have to be tested further because this was a single-blinded experiment. It was phase 1 and 2. Phase 3 trial would have to be performed to see if it still functions, if it can be used as a vaccine. But this, for now, it looks like very promising. It doesn't have massive symptoms. It has mild to medium symptoms. Safety-wise, it looks good. So this looks like a promising vaccine. Another vaccine, which I would like to talk about and has been in news a lot after its successful phase 2 trial, is from Mrna company, pharma, and it is mRNA 1273. The way it's done is you have the RNA and you use a PCR machine, and basically in the vector, it could be expressed, and then it is provided in the form of vaccine. So here you have, for example, in the cloning, you have a vector, and then it expresses mRNA, and there is a capping performed, and I can talk about that more if needed, and that is used as vaccine. Now, when it goes in, the mRNA would be expressed, and that would elicit response, and that is used as a vaccine. And so far, phase 1 and phase 2 trials look really promising, and phase 3 trials are approved. So, but the race is still on. There are many more vaccines in the trial process. But looking at the next steps in the current situation, what can be done if we wanted to open the country for trade again, if we wanted to get back to the business completely like it was before. So some of the governments are suggesting that having antibodies to SARS-CoV-2 and checking the presence of antibody would probably be a certificate to again get back to work. Because that would tell that you probably were able to fight before and you probably have antibodies for it. Maybe your body is able to have them, some memory of it. And in case of SARS, it has about 1 or 2 years of memory. So possibly for SARS-CoV-2, that would be the case, but it's not confirmed. However, increasing evidence is showing long-term or lack of long-term immunity for this infection. So we do not know how useful this method would be. So there is a lot of unknown information. There are a lot of unknowns related to SARS-CoV-2 in terms of vaccines. We do not have a definitive vaccine. We do not have a definitive drug yet. Vaccines are used in combinatorial with other vaccine. But we still have to figure out a lot of things as to what would work, possibly. With that, the race is still on. We would have to figure out what next. And that's all I have for now. So I would be open to, happy to take any questions.
Madam, please unmute yourself. Shubha, madam, please unmute yourself.
Thank you, sir. Good morning, everyone. I'm Dr. Shubha Diwan from St. Thomas College, Bhilai. Firstly, I would like to thank Manjula ma'am for accepting our request and delivering such a enlightening talk about the molecular aspects of binding of this virus. We can see from the chat box itself, ma'am, how interesting your talk was. Thank you, ma'am, once again. Ma'am, some participants have dropped some questions in the chat box, and I would like to convey the same. Ma'am, one of the participants has asked that as in India, we can see that death rate is slightly slow, comparatively low. So does it have any connection with the innate immunity?
It is hard to say. There were reports that the virus that's infecting in India is slightly different based on the type of mutations it has. SARS-CoV-2 is different compared to what we have in other parts of the world. But how important that aspect or how it affects the infectivity or our immunity is hard to say. We do not have any definitive answer to that yet. Is it our innate immunity? I don't know about that because we cannot answer that question. Is it because the type of environment we live in or the food or what is causing that? We do not know. So I think we would have to figure a lot more things before we say that.
More researches are needed for this.
But the other point is there is a lot that we can say about the way India is handling currently the situation. So there was a massive lockdown where people were not allowed to go out of houses unless it's necessary. That was not the case in many other parts of the world. For example, I live in New York, and we did not have that situation. So people were able to commute around, and that's what caused the infection. And then because of the way we have commute, everybody has to, in New York, we have to use public transport. There is no, the parking system is very different. You cannot just park like other parts of the country because of the space limitations. So most of the people use public transport. And also we did not have like the way in India, it was sure shot regulated that you cannot go out. That was not the boundary here. We were told that it's up to you if you want to go out, but stay in, wear mask. Mask has become more mandatory now, but it was not the case initially. So, a lot of things, the way India handled it was really well, but it would be a situation now that the India has opened for trade again. Then we can, maybe with more testing, we can answer that question.
Thank you, ma'am. And one more question is there.
Definitely.
One of the participant has asked that, is there any relation between the action of ACE receptor protein and the appearance of symptoms in this case of SARS, COVID?
Can you repeat the question, ma'am?
Ma'am, is there any relation between the action of ACE protein and the symptoms which are appearing?
Action of ACE protein. So
working of ACE protein, I think.
So ACE2 is a receptor to which spike protein would bind.
Okay.
And so there was a slide that I talked about on the affinity with which the spike protein would bind to the ACE receptor on human cell. Now, it's an interesting question, how affinity or how tightly it binds, how does it affect the infection process? It is not completely known.
Okay.
But we can say that there is a higher affinity that we see compared to other coronaviruses, and which is why we see a lot of infection compared to other coronaviruses.
Okay, ma'am. Thank you, ma'am. We have a lot of questions, but as we are time bounded, we have to wind this session.
Yeah, definitely.
Thank
if
you, ma'am,
I'm sent those questions, I would be happy to send the answers to you.
Okay, ma'am, sure.
Okay.
Thank you, ma'am, once again for giving such an interesting and minute details about the molecular aspects of this virus and the immunological response against it. And now I request Shanti, ma'am, to continue the session.
Thank you, Shubha, and thank you, Dr. Manjula. Now I move on to the second speaker. A second speaker for today's webinar is Dr. Harsha Ramchandani. Dr. Harsha, who is an MD, is the Chief Medical Officer at Bay Area Community Health located at Fremont, California. Dr. Harsha joined this health center in April 2015 as a clinic director. She is a board certified internist with over a decade of experience working in community and outpatient settings within different communities of Michigan. She did her MBBS degree from Gandhi Medical College, Bhopal, following which she completed her residency in internal medicine from the same Gandhi Medical College, Bhopal. Before joining Bay Area Community Health, she has earlier worked with Sinai Grace Hospital and was also affiliated to Detroit Medical Center. Dr. Ramchandani has also served as an adjunct professor for American University of Antigua Medical School. In her current position, she supports the medical staff in monitoring of medical policies and procedures and also provides clinical guidance and oversight related to medical care for all providers. So, Dr. Harsha Ramchandani, being a doctor by profession, will give a talk on the epidemiological aspects which probably will make us understand the reason behind the high infectivity rate of COVID-19. Dr. Harsha, please.
Thank you, Dr. Shanti, for such a warm welcome. Actually, I'm going to share my slides here. Okay, let me do that. Okay, can you all see my slides?
It's not shared, ma'am.
Oh, it's not shared. Okay, let me stop sharing and do it again. 1 second. Share. Okay.
Yeah,
How about now?
Yeah.
You can see
it?
You
can make full screen, madam.
Can you see it?
Yes. Thank you, ma'am.
Okay, very good. Well, good morning, everyone there. It's my pleasure to present in front of this bright class of microbiology. And I'm really excited about this. Thank you again, Dr. Shanti, for giving me this opportunity. to share some of the experiences that we are facing here in United States related to COVID-19. But as Dr. Shanti has mentioned, I just wanted to keep one slide for introduction of myself. So I am, as Dr. Shanti mentioned, working as a chief medical officer. But as you can see, it's highlighted. It's a small font, so you might not be able to see it. But I did my education from Bhopal. It's a Gandhi Medical College. I did an MBBS, and then I did an MD also from Bhopal. And then after that, I did some education in Detroit Medical Center. But currently, I'm in California, as you can see on the corner on the Pacific side or the west side of United States. And this is the Golden Gate Bridge, which is famous for the San Francisco in California. And that's where I'm working as a chief medical officer currently. But my grounds are back in Bhopal, Madhya Pradesh, and that's where I am from. So I'm going to speak in both Hindi and English right now because I'm proud of that. I'm very fluent in Hindi also. And I was listening to Dr. Manjula's talk a little bit, which was very wonderful. And I am going to add a little bit of clinical piece in this because, as you all know, I'm working as a clinician right now. So I'm seeing the patient side of it also. We're seeing the patients, we're testing the patient right now, and we're treating them also in our clinic. So please feel free to add your questions in the chat box. Hopefully, at the end of the session, we will have time to answer. I will have time to answer some of the questions. Looking at the time, Dr. Shanti, it's 10:25. I started at 10:25, and I've given 25 minutes, 10:25 of my time, which is, so I have 25 minutes. So I'll end at 10:50, right on target. So let's go to the next slide. We have the contents, and that's what I'm going to talk about is epidemiology, virology and transmission, clinical manifestations and diagnosis, antibody and immunological response, especially related to the clinic's situation. What do we as a doctor do about that, and the treatment and prevention. Okay. So the first thing first, like we are calling it SARS-2 or COVID-19. So ये नाम कहाँ से आया हमारे पास? Like, why are we calling it SARS-2? So SARS-2, basically, it's a short form for, as you can see here, it's a severe acute respiratory distress syndrome. And why we're labeling it as 2? Because as you all might have heard from Dr. Manjula's talk also, and I know this is a bright class and we have all the information now on the online. So I already imagine that all of you have a lot of information. So SARS-1, we don't call it SARS-1, but just SARS, that happened back in 2002, which was a similar coronavirus that also caused the pneumonia or the severe acute respiratory distress syndrome. So, oh, okay. I don't think you are seeing my slideshow in the full format here. Is that right? Let me just try doing that again then. 1 second. Okay. So I click here. Okay, it's still seeing like that. Okay. I have 2 screens here. Maybe that's why. Okay, 1 second. Sorry for that. Okay, I'm clicking on the slideshow, but it seems like it's not. Oh. Somehow you're seeing my next slide also there, I guess, but that's okay. We'll continue like that, I guess. Is Dr. Shanti, or can you see, like, is it okay? Or should I try more? Let's just say zoom into the slide here.
It's fine, you can continue.
Okay. Yeah, I have 2 screens actually, so I think it's projecting on the other screen. Let me take out that screen once. Maybe. Okay, and then let me click on the slideshow. Okay, that's perfect. Yeah, I have 2 screens connected. That's why. I think that's better, right? Okay, I hope it's better.
That's fine, you can continue.
Okay, but do you see the full screen slideshow now? That's what I'm seeing right now is a full screen slideshow.
Yes, madam, you can continue. It's fine.
Okay, very good. So I was telling that SARS is a severe acute respiratory distress syndrome, and why we're calling it as COVID-19. Again, it's a short form: CO for corona, V for virus, D for disease, and 19 is for year 2019. That's when it was started. It's all started. So it's all COVID-19, a short form. And we all know it's spread easily from person to person now. So focusing a little bit on the epidemiology, because that's an important piece to know where it started, how it started, and how we could have prevented it, or how we can prevent it right now by looking at the immunological response of the body and talking about the creating the vaccines or developing the vaccines related to that. So again, this is one of the slides that's showing, and this is the point where it's not a surprise now. Everybody knows it started in Wuhan in December of 2019. So there was a pneumonia of unknown cause that was in few patients in the hospital in the Wuhan. They saw patients which were being admitted related to the pneumonia, and they didn't know what's the cause of that, and that's how finally WHO was informed of that. And we found out about it. So there are sequence of events right now that I wanted to just go over that quickly. But again, as related to this class, I wanted to focus a little bit. So the 31st December, that was the first official report of China was shared with the WHO. And that's when on January 2nd, WHO started the investigation. And the main thing that I wanted to share was on January 10th. So it was pretty fast. And I think the delay was informing the WHO. I think in Wuhan, whatever cases happened, they were suppressed. They were not informed properly to the agencies or to the government. And then once they were informed to the WHO on January 10th, that's when the virus was genetically characterized. So we are learning a little bit more about the cases, the first few cases. So the first 425 cases we saw, and we realized that, as you can see, this orange color is the cases which were linked to the Hunan market, and the pink color is the one which is not linked to the Hunan market. So you can see a lot of the cases were still discovered, people who didn't go to those markets, and they were still diagnosed with this. And the PCR diagnostic reagent was developed and testing was completed by China and CDC quite in the time of January, February. So again, as I said, I'm a clinician. I see patients on a day-to-day basis. So I wanted to share this slide about the disease burden. Kitna COVID-19 is affecting our population on a day-to-day basis. And I have presented a similar information to another college like a month ago, and I can see the cases have doubled. Just rate से ये cases बढ़ रहे हैं, which is very very concerning. And हमें ये देखना है कि हम इस चीज को कैसे prevent कर सकते हैं. तो इस talk से मैं एक चीज और मेरा goal है, of course, I mean as a student, you all are learning about immunological responses and microbiology and other things. But another thing, as a human being, as a civilian or as a citizen of India, हम इस presentation से कैसे और चीजें learn कर सकें जिससे कि हम इस चीज को prevent कर सकें या अपनी family and friends को educate कर सकें इस चीज से लेके. तो उसके लिए आप disease burden अगर देखें आप, तो it's just a comparison United States versus India. But again, as I said, my roots are still, they are from India. So I'm following it very closely and my family is still in India. So 1,639,000 cases as of now, and again, this is from July 31st, like today, this morning, the latest update is this, and there are 35,786 deaths right now in India, which is very unfortunate. In United States, actually, the thing is even worse than any other countries, which is, you can see 4,634,000 cases diagnosed so far, and there are 155,000 deaths so far in United States. And it's a major, major problem. We have not encountered any of such problems in the last 100 years. So it's really big issue. So we talked about coronavirus a little bit initially, like it causes SARS, and but corona is a Latin word that basically says that it's a garland or the wreath which is surrounding this virus. And it's an envelope positive strand family of single-stranded RNA viruses, which is causing the infection. Now, there are 7 types of coronavirus that can infect human, and 4 of them are very common, and they cause the common cold-like symptoms. But 3 are rare, and they can cause severe respiratory syndrome. And this coronavirus or COVID-19 we're dealing with is that type of uncommon virus causing problems. Again, it's very unfortunate. And as you all know, I mean, of course, we have been talking about person to person. You know, you have to wear masks. If anybody has symptoms, they have to wear a mask. It's a universal thing. Sabko hi mask pehenna hai. But apart from that, even if you're not wearing, if you're not in contact with somebody direct, person-to-person contact, still there have been cases identified. तो वो cases क्यों होते हैं? तो जैसे आप ये picture में देख रहे हैं कि सामने वाले ने sneeze किया, तो सामने if somebody is there, they can get the infection, of course. But even if there's nobody there, these small droplets, they accumulate on the surface. And if somebody comes in and then touch those surfaces and then puts those hands in either the mucous membranes like the nose, mouth, and eye, and that's how the virus can enter the body. तो काफी सारी स्टडी हुई है क्योंकि नोवेल कोरोना वायरस है, किसी को पता नहीं है इसके बारे में कि कितना कैसे ट्रांसमिशन होता है शुरू में, कैसे किस चीज से होता है. सो वी आर ऑल लर्निंग एज वी स्पीक आल्सो, बट लॉट ऑफ स्टडीज हैव शोन कि कार्डबोर्ड पे है तो कितने घंटे वायरस रहेगा, इफ इट्स ऑन द प्लास्टिक हाउ लॉन्ग, इट्स ऑन द कॉपर और स्टेनलेस स्टील हाउ लॉन्ग. तो देयर हैव बीन स्टडीज दैट हैव शोन दैट द वायरस इज अलाइव on the surfaces for a couple hours, which can range from 8 hours to 80 hours. So what we say here in United States, like if you get a packets from Amazon or from anywhere, UPS, so packets जो आते हैं उनको आप गराज में ही रखें या बाहर रहने दें थोड़ी देर के लिए, या ग्लव्स पहन के उनको अंदर लेके आए, क्योंकि जो पैकेट्स रहते हैं उस पे भी वायरस हो सकता है. So it's something to consider, and definitely the public transportation. अभी तो इंडिया में सब बंद है, especially मध्य प्रदेश and all, but still public transportation, if you go or if you go to a public places, before touching the surfaces, just be a little careful about that. So again, this is how it's just a figure showing how the disease is progressing fast, and this is the virus with the reproductive number of 2. So one person affects 2 people, and then finally it affects 4, and then 8 and 16, and that's how the reproductive age increases the transmission of the virus. So again, we just, as a doctor, as a clinician, we want to make sure we cut the cycle, we break the cycle somewhere in between, so that the person which is affected or infected does not transmit the infection further. So contact tracing is a very, very big thing that we are currently spending time on. So contact tracing में ये होता है कि जैसे एक patient कोई positive आया, हमने clinic में test किया and that person is positive, तो यहाँ पे जो Department of Public Health होता है वहाँ पे हम inform करते हैं, and then we also call that patient and then find out कि आप किस किस से मिले थे. तो अगर वो बताते हैं कि हम लास्ट 10 days में इतने लोगों से मिले थे, तो हम उन लोगों को भी कांटेक्ट करते हैं एंड देन बोलते हैं कि यू स्टे होम फॉर 14 days, क्वारंटाइन योरसेल्फ, so that we can contain the disease right there, so that it doesn't spread further. But again, it's a new concept. We're all learning and trying to educate our patients. Again, then you might hear a lot about incubation period. So incubation period is actually the time between the exposure to a virus to the onset of symptoms. And that usually can vary from 2 to 14 days, but the majority of cases aate hain, they come from like on the 5th day. So from somewhere from 4 to 6 days of the exposure to the virus, but still the incubation period is 14 days. And patients are most contagious when they're symptomatic, of course. When they're sneezing and coughing, they're going to be more symptomatic. So a lot of terminology we use right now, and I am sure you are learning also and reading online about these things. And right now we say it's a community spread disease. It's not just related to the Hunan market or not just related to one thing, but it's a community spread also. So even when the people, they don't know if they met anybody with symptoms, they're still suffering from this disease. And we think it's all related to the droplet transmission. Social distancing करने को बोलते हैं आजकल. Everybody is very aware, especially everywhere in India also. Social distancing रखें आप. कोई भी congregate settings होती हैं जहाँ पे भी एक साथ मिलने का grouping होती है. अभी काफी सारे festivals भी आ रहे हैं इंडिया में. So just try to prevent any gathering, too much gathering. क्योंकि अगर एक भी patient वहाँ पे, अगर एक भी आपका friends or family member is asymptomatic right now, of course if they are symptomatic they're not going to come out. But if they're asymptomatic, they can still transmit the infection. So we try to reserve that groupings as much as possible, and maintain the social distancing of 6 feet and wear the mask. And isolation, what is isolation? That's basically to separate those who are sick with a person who is not asymptomatic. So the person who is sick staying alone for 10 days, that's called as isolation, whereas quarantine is basically the person who is not sick but they just got exposed to someone, and we say the person should quarantine themselves for 14 days. So again, quarantine means the person is not sick or not positive, but isolation means the person is positive, and that's why they're isolated out for at least 10 days. And of course, this is a big pandemic that we're talking about because the disease has spread all across the continents. So going to the immunology, what are the antibody responses that we see for SARS-CoV-2? And this is very helpful for us to test to understand how to test our patients, or what are the tests that we do, and which I will show in the next couple slides, like how we do the testing in the clinics and what are the tests that we are doing right now, but how the antibodies are developed. And again, this is a study that we are trying to. I'm sorry. Did I stop sharing? Oh, let me stop sharing again and then share screen again. 1 second. Sorry. Okay, there you go. So the antibody response, how it happens to the COVID-2, and these are the days after the symptom onset. And we look at the IgG and the IgM antibodies. IgM are the acute antibodies that happens when the patient has an acute infection. And IgG happens, the antibody which is after the infection is over, and it's the antibody which shows a recovery from the COVID virus. So these are the days that you can see, these many days it takes for the antibody response to happen. And these are the levels, how much is the level of antibody that is seen after the symptom onset. Again, this is the IgG and this is the IgM antibodies. And these are the other things. Again, I'm going to focus more on the clinical symptoms. There are clinical signs that we see as the time progresses from COVID-19. And that's how the potential therapies are also related with that. So we say there are different stages of COVID-19. The first stage is called as a viral response phase. The second stage is the pulmonary phase. And then the third stage is the host inflammatory response phase. So sometimes we are seeing patients jo ki sick hote hain shuru mein, and then after a couple of days they feel fine, and then they don't develop the pneumonia or the pulmonary phase. But then after some time, after being normal, feeling normal for some time, then they develop again an inflammatory response. So that is the stage 3, which is related to the hyperinflammation phase. That's also a serious problem that we are seeing right now. And then as I've seen, the potential therapies are related to that, which I will also present in the next couple slides. So again, as a doctor, we see patients, and काफी सारे patients जो हमें call करते हैं, वहाँ पे हम उनको mild symptoms के लिए बोलते हैं कि आप घर में ही isolation करें और घर में ही आप जो भी अपना conservative treatment बोलते हैं हम वो करें. But most common symptoms जो होते हैं वो है fever, cough, shortness of breath, and the loss of smell and taste. And other symptoms can be as listed below, which are here. So right now, what we are doing in our clinic is we're checking everybody that comes, including the staff. So जितने भी doctors और nurses हमारे clinic में आते हैं काम करने के लिए, उनको morning में हम उनका temperature check करते हैं, and then we record the temperature. And then because the fever is one of the common symptoms, so we try to do that. But हम also हर महीने में एक बार अभी अपने staff की testing भी करते हैं clinics में. And of course, if there is an exposure, then we definitely test them, all of them also, just to keep it safe. And of course, all the patients are all tested also for temperature. And then the severe symptoms of COVID-19 or the emergency warning signs होते हैं, which हम educate our patients and our nurses, is difficulty in breathing or shortness of breath, which can suggest the ARDS as well as pneumonia, or या फिर persistent pain or pressure in the chest, new confusion, and bluish lips or face. These are definitely very, very serious symptoms that we keep an eye on. So now another thing that I wanted to see, again, as a member or as a person who is suffering the pandemic already, I mean, it's might not related to your course or immunology or microbiology, but still थोड़ा सा clinical symptoms हमें पता होना चाहिए कि COVID-19 में क्या होता है versus common cold में क्या होता है, क्योंकि अभी especially season change होगा, सितंबर अक्टूबर में, तो काफी केसेस ऐसे भी आएंगे जो कि कॉमन कोल्ड हम जिसको बोलते हैं कि नजला हो गया करके। तो कैसे हम कोविड-19 और नजला को डिफरेंशिएट कर सकते हैं? Although a lot of symptoms are very similar, but still this is something, that chart, which I really like it, and I share with my patients also to understand and remember this. There's definitely some immune effects जो कि हम labs वगैरह check करते हैं, जैसे ल्यूकोपीनिया और white cell count कम हो जाता है कोविड-19 में, थ्रोम्बोसाइटोपिनिया या प्लेटलेट काउंट भी कम हो जाता है। बट द मेन थिंग जो आप इसमें देखेंगे कि जो नज़ला कॉमन कोल्ड होता है उसमें आपको ज्यादातर रनी नोज महसूस होता है, इन देन सोर थ्रोट हो सकता है, बट वर्सेस कोविड-19 में द फीवर इज वन ऑफ द मोस्ट कॉमन सिम्टम्स, and then cough, which is mainly the dry cough that happens in the COVID-19. So again, as an educated citizen and student, it's important for you to educate your family and friends also. So please keep that in mind. And then loss of smell and taste has been, you won't believe it, so many of my patients, they come in and they tell that they have loss of smell and taste, which is typical symptom of COVID-19 nowadays. Some people can also have diarrhea, nausea, vomiting with COVID-19. So how do we do testing for COVID-19? And next slide, I will show how we do it in our clinic. But standard of care we have, and also in India, I speak with a lot of my friends. Hamidia Hospital, the main and Chirayu Hospital in Bhopal, they have a COVID-19 ward, and that's how they also do testing there. And we do it by RT-PCR. And the samples we collect from nasopharyngeal, oropharyngeal, mainly in the clinic. And alternatives testing होते हैं, IgM ELISA भी अभी आया है, which is a point of care testing, जो कि जिसमें टाइम नहीं लगता है रिजल्ट आने में. RT-PCR का जो रिजल्ट आता है, that still takes 1 से लेके 3 दिन तक लगते हैं रिजल्ट्स आने में, जबकि जो IgM ELISA टेस्ट होता है, that comes really fast. So this is a machine that we just bought it recently in our clinic. And the name of the machine is Sophia, that gives the results in 15 minutes, and that checks for the antigen in the blood. So it's not as sensitive as PCR, but again, it's a 15-minute test. So we really love that, actually. And these are the indications where we test in, right now, the place where I'm in California, in Alameda County, we test each and every person. There's no, anybody can come in and we test them. There's no problem with that. But I know in India, they test only when you have symptoms or you have the exposure to COVID-19. So this is how we do drive-through testing in the cars. That's what we do in our clinic. So आप अपना बस window आप नीचे करते हैं, and then patient is sitting inside, and the recommendations are to do it outdoor in the where there is air circulation is good. And actually, this is me doing the testing, and this is my nurse who's helping me. So we wear all the PPE or personal protective equipment to do the test. We just collect the sample quickly, and then the patient leaves. And then we get the results in 2 days, and then we call patients with the results. And we are seeing a lot of positive patients in our clinic, unfortunately. So quickly moving on to treatments, and I will aim to close my session by in like 5 minutes. The treatments, what we talk about, and again, I know online information जो रहती है, कई बार गलत भी information बहुत मिलती है. And there are some politicians that raise the information, which as a clinician, as a scientist, all of you should take with a pinch of salt. So कभी भी आप कोई article पढ़ते हैं तो उसमें आपको देखना चाहिए कि randomized trial था कि नहीं था, इसमें कितना controlled था कि नहीं था, placebo group था कि नहीं था. So unfortunately, Trump ने hydroxychloroquine को बहुत famous किया है, not just in the US but in India also. But that's not the drug which has shown much of the benefit right now in COVID-19. So unfortunately, hydroxychloroquine we're not using at all. Azithromycin is an antibiotic that can help a little bit with pneumonia, but not much of the help with the COVID-19 SARS symptoms either. I will spend some time talking about favipiravir because India is actually leading the trial for favipiravir and antiviral. And then, of course, IL-6 inhibitors, corticosteroids. There is a drug, remdesivir, that we are currently using right now in the United States, but only for a very sick patient in the hospital. And then, of course, plasma from a recovered person from COVID-19, and then IVIG are the other treatments that we could use. So as I told you about favipiravir is an oral antiviral, and it is already approved in Japan for influenza, and it selectively inhibits RNA polymerase, which is necessary for viral replication. So India में Massachusetts यहाँ पे भी Harvard College में इसका trial चल रहा है, but India is already ahead of that, and a phase 3 trial is going on right now, which started in May 2020. So I was just recently told by one of my colleagues, he's a pulmonologist in India, that this is available right now in India for treatment, and the cost is also not too expensive, and it is available right now for COVID-19. So just for your knowledge, again, it's information for your friends and family also. If there anybody sick in the hospital, favipiravir is available in India. Interleukin 6, one of the slides I showed you for antibody performance. So interleukin 6 is actually an inflammatory cytokine which is produced by cells like lymphocytes, monocytes, and fibroblasts. So SARS infection जब होता है तो interleukin 6 का production बढ़ जाता है, which can cause more harm than the benefit in this situation. So there was a trial, there was a drug called serilumab, which is an inhibitor of IL-6, but it has not shown much of the benefit in COVID-19. And then there was a UK recovery trial which showed that the low dose of steroid, which is dexamethasone, can be helpful. But again, as I say, we have to take the studies with the pinch of salt, or we have to read the studies and understand that. So no benefit was seen in patients who did not require respiratory intervention. So ऐसा नहीं है कि अगर mild symptoms हैं आपने steroid दे दिया and that will be beneficial. It's not actually. So again, how can we prevent it? That's another important thing, because this is a pandemic. We are all suffering from this, and a lot of scientists are working on developing the vaccines and developing the antiviral medications. But as a informed citizen, how we can help it? So again, I'm going to emphasize this highly on this. This is the most important slide of my presentation. I'm going to say, be informed, be prepared, read the material from a reliable source. Don't just go online and India newspaper jo de rahe hain aap news usko padh ke agar usko maan lenge to that's not going to be a good thing. Read the articles properly. Maintain the social distancing, 6 feet. Wash your hands frequently. Sanitizer nahi hai aapke paas, koi problem nahi hai. Aap hand wash karna bahut zyada important hai, and 20 seconds washing hands is important. Avoid touching your eyes, nose, mouth. Stay home when you're sick. That's another very, very important thing. And I'm hearing from a lot of people in India कि बीमार थे, ठीक हो गए, 3 दिन में अभी बाहर चले गए हैं. तो 3 दिन में नहीं होता है, आपको 10 दिन के लिए आइसोलेट करना जरूरी है अपने आप को, अगर आपको कोई भी सिम्टम्स हैं. Even if COVID test was not done, if you have a fever and cough, it's COVID-19 unless proven otherwise right now. So please educate your friends and family members. Anybody sick, 10 दिन के लिए एटलीस्ट आप उनसे दूर रहें और उनको बोलें कि वो भी दूसरों लोगों से दूर रहें. And of course, use the mask, every day. And I think I just have 3 more slides. Just a little laugh. I just want to, again, mention about emphasizing about the mask, wearing the mask as one of the things. So this slide actually says, and as you know, Trump is notorious and he does not wear mask. He just started wearing mask recently. So Trump has been advised by staff to wear a mask, but nobody told him what type of mask. So he's wearing a Batman mask here. That's not how we want to do it. Antivirus CD aapne laga li. I mean, it's again funny. Wearing the mask properly, that's how we want to show in these 2 slides. Another important thing, as I also see patients as a diabetologist, I see patients with diabetes a lot. And I'm seeing a lot of patients because of coronavirus, they're at home and not exercising, and they're gaining a lot of weight, and the diabetes is out of control. So this is Monica from Friends, if you remember that series. This is me before quarantine and me after quarantine. So, I mean, it's funny, but at the same time has a message that please exercise, please stand up, please walk outside, wear a mask, and please do some activity even at home. And also tell your friends and family members, especially if you have parents with anybody with diabetes and hypertension. It's a very important thing. And don't just keep watching TV. That's not good for your eyes. But this is an important slide that I wanted to share. Vaccines, as Dr. Manjula also mentioned from Mrna, there is a vaccine which is already in the phase 3 trial. There are 30,000 clients or the people who got the vaccines. They're still enrolling in the study, and there is a lot of hopes from that vaccine from the Mrna, and a couple of trials are going on, but it's under construction. It's going to take a couple of months, at least the end of the year. We are hoping to get it by the end of the year. So what's next? The last slide of mine, and I'm gonna say that again, as a doctor, I see patients, and there's a lot of stress going on, a lot of anxiety going on. Like, what's gonna happen with COVID-19? कब ठीक होगा? कब हम बाहर जा पाएंगे? Especially young age के students बोलते हैं, कब हम अपने friends से मिल पाएंगे? कब हम classes कर पाएंगे? A lot of teachers are also concerned कि online teaching is hard for them. कब हम खोल पाएंगे? So everybody is going through the same question. I'm gonna say, stay calm. Learn new things, catch up on the college work, spend quality time with your family, and stay healthy. That's the most important slogan for this year. Stay healthy. This will pass. Everything passes. This is also going to pass. So believe me, and stay healthy and stay calm. Thank you very much. I'm going to stop sharing now. This was my last slide.
Thank you so much, ma'am, for sharing this information. As you're on the end on which patients are being treated, and therefore you can very well explain this epidemiology of this virus. Ma'am, here are some queries from the participants, and I wanted to convey the same. Now, one of the participants has asked that many people are asymptomatic even if they have infection. So how it is caused?
So again, everybody has a different immunity. So we are all learning this. So especially the young patients, and that's the reason why the spread is going on so much, because the young patients have a higher immunity, so they can fight the infection well. And versus the older elderly population, in which we are seeing the more complications like pneumonia. So that's why, the virus is same, but the immunity, the body immune response is different. And the young people, they don't realize, and they get the infection and they pass it on to their grandparents or the elderly seniors in the house. And that's why the problem has been happening. And here in United States, especially, we are having a hard time controlling. जैसे डॉक्टर मंजुला ने भी बताया कि इंडिया में ज़्यादा कंट्रोल है कॉप्स का एडमिनिस्ट्रेशन का. In United States, it's not like that. We never had a complete lockdown. Kids are still going out to the beach and playing basketball, but they're asymptomatic. To answer your questions, it's mainly because of their immune response.
Thank you, ma'am. And one more question is there. Ma'am, we are very much hearing about herd immunity regarding this infection, herd immunity. Yeah. And ma'am, does it have any role on this pandemic?
I definitely think that there is a role of herd immunity in this pandemic. Because the people who have had the COVID-19, we are studying those population to see, and we are actually getting the plasma from those populations. They have antibody to see if that helps. So a lot of studies are going on right now to realize and to see if the herd immunity is going to help prevent this infection in future, or even right now it's going to cut the cycle. But again, as you know, the population of the world is a lot. And right now, Dr. Anthony Fauci, who is the infectious disease doctor here, has said that 80% of the population in the United States will get infected, and that's when the herd immunity is going to be beneficial. So it's a big number, but we are hoping that it's going to be beneficial for us. Now, another concern is about coronavirus changing its mutation next year, and if the patients have herd immunity right now, or the people have herd immunity, what's going to happen next year? Which is also questionable right now, but we are hopeful. I'm hoping for the herd immunity for sure.
Okay, thank you, ma'am. And one more last question.
Sure.
Ma'am, could you throw light on the aspect of therapy currently being used for treatment of this virus? Is it a replication suppressor or anything else?
I'm sorry, replication of?
Replication suppressor. Anything that is suppressing the viral replication, the therapy, the medicines which are currently being used, are they suppressing the replication, or they are acting on the symptoms, or like that?
So that's a great question. So different medications are acting in a different way. So remdesivir and favipiravir that I mentioned, they act on the replication, and they prevent the replication of the virus. But then I did mention about the dexamethasone, which is a corticosteroids. It does help improve the inflammation. So it's an anti-inflammatory medication. So wherever the virus is causing inflammation in the lungs, it causes the pneumonia. So it helps prevent that inflammation in the lungs, and that's how it helps. So it's both ways, equal way. And then antibiotic azithromycin, it helps prevent the superimposed pneumonia related to bacterial infection.
As you told that Vepiparavir is replication suppressor, but it is in third stage trial. So nowadays the antibiotics are mainly used, I think, for
So,
suppressing the infection.
yeah, it's in the phase 3 trial. Started May 30th of this year. But I was again told by one of the pulmonologists, and this is a confirmed information, that he is treating patient with favipiravir in Bhopal. So it is available. FDA or the institution in India, they have already approved it on an emergency basis. So it is a medicine which is being given, but not to the everyday patients, but only to the patients who are in the hospital. So they are sicker patients.
Thank you, ma'am. Thank you, ma'am, once again for throwing light about the spread and epidemic of the treatment of this pandemic. And the participants are definitely benefited from your talk. Thank you, ma'am. Thank you so much. I again
Thank you,
request
Dr.
Shanti,
Diwan.
ma'am.
Yeah, and
Yes,
thank you
ma'am.
to you also for arranging this along with Dr. Shanti and Michael. Thank you.
Thank you, ma'am.
Okay,
I
I'm
again
gonna
request
go.
Shanti, ma'am.
Yeah, I'm gonna go, Dr. Shanti. I'm gonna leave this now, and please feel free to email me if there are any other questions.
Yes, ma'am.
Sure, madam. Thank you so much
Okay.
for your talk. Thank you very much.
Okay. Thank you, all of the students here. It was nice seeing the energy in the chat box as I was looking in the comment section. So thank you, and you have a great faculty here. So I wish all of you a good luck for your future. Bye bye.
Thank you so much, ma'am. Thank you, Dr. Shiva, for the queries put up in the chat box by the participants. Now we proceed on to the third speaker of our webinar. We now have our third speaker of the day, Dr. Sunil S Porad, who is working as a scientist in bioinformatics at Bioresources Database and Bioinformatics Unit, which is affiliated to Institute of Bioresources and Sustainable Development, Imphal, Manipur. Dr. Sunil has completed his master's in biotechnology from Amravati University, after which he proceeded for advanced diploma in bioinformatics from Bioinformatics Center, University of Pune. He then proceeded to complete his PhD in biotechnology from Sant Gadge Baba Amravati University. As for his awards and honors, he was sponsored by DBT for his diploma course in bioinformatics. He has also availed a travel grant to participate in international conference on bioinformatics at Hong Kong in the year 2007. Dr. Sunil, as a coordinator, has completed 3 projects funded by DBT and 5 projects as co-investigator, of which 4 were funded by DBT and 1 from ICAR. He has authored 5 book chapters and has 16 publications to his credit in peer-reviewed journals. Presently, he is working on the area of biological database, biodiversity informatics for the past 17 years. Dr. Sunil, being in the profession of developing databases, we are now anxious to hear, sir, from you what's new related to COVID-19, how these databases which you have developed recently are being considered for the development of vaccines, etc. Dr. Sunil, please, sir.
Sir, please unmute yourself. Dr. Sunil, please unmute yourself.
Thank you, madam, for the introduction. I thank for my talk. Today, I will be talking not data, but the topic for is bioinformatics, like how can come back to corona. So, I share my
Sunil, sir, your voice is breaking. It's not audible.
Is it audible?
Pardon, sir.
Hello. Am I audible?
Sir, your voice is breaking.
Hello.
Hello.
Yeah.
Yes, sir. Please continue.
So, I'll be talking about bioinformatics studies in corona. First of all, I tell you that uh, I I was expecting that some sort of similar today, and I'm thankful for the speakers who have really uh, spoke more uh, in different views. I biochemistry and medical field. So, my topic is different, and probably will be covering right from the beginning that how what the point to study. So, let me start classification of the study of virus in
Sir,
class.
sorry to interrupt you. Sunil, sir, sorry to interrupt
Yes,
you, but
yes.
your voice is not audible.
Mm.
You're not audible, sir, please.
Shall I stop my?
Sir, please
Is it
try
audible?
without headphone.
Yeah. What is there? Shall I?
So your voice
Okay.
is still breaking. Please use without headphone. I guess that will work.
Is it audible now? Am I audible?
Hello.
Yes. Am I audible now?
Yes, sir. Yeah. Now you're audible. Please continue.
Okay. So, uh, talking about the classification, this corona belongs to order nidovirus with family coronavirus, and there are few genus and species names are written here. First was human coronavirus 229, human coronavirus 43, human coronavirus NS53, human coronavirus HKU1. Then this SARS coronavirus was there, severe acute respiratory syndrome coronavirus, human interleukin coronavirus, and another was torovirus, to which species human torovirus. Sorry. About the structure and replication of this particular coronavirus inform. It is positive and single-stranded RNA, as you all know. Its genome size is around 30,000 nucleotides long. It is pleomorphic to axis, uh, having size of around 80 by 160 nanometers in diameter, with 12 to 24 nanometer surface projection spikes that cause the and appearance that like the brown, so the name that's given coronavirus. So the major proteins are spike proteins and the membrane proteins, and you can see some of the itself. Just to give a history about the coronavirus, this coronavirus was defined as viruses of, as I said, 120 nanometer diameter with a crown-like appearance first described in 1930. With the first coronavirus, HCO-229, it was isolated from humans in 1965. The name, as I said, was based on structure, crown structure, and based on their antigenic relationship, coronavirus was classified into three groups. Group 1 was with HCOV-79 and HCOV-NS53. Group 2, HCOV-43 and HCOV-SQ1 is composed of mammalian coronaviruses, and group 3 was avian coronavirus. And as you know, as we are talking more about the coronavirus, the present coronavirus, SARS-CoV-2, and this SARS-CoV is an outline of SARS-CoV-2, although some phase 2. So we're talking about the biology also, like to see where it actually uh, stands. So as you can see, the red arrow shows the group 1, which belongs to one coronavirus. green arrow, the coronavirus which belongs to group A, that is COV, OC43 and SQ1. And as I was telling that subgroup B is the beta coronavirus, that SARS-CoV and SARS-CoV-2. Now, they are actually outlined. As you can see from biology itself, that they are not with the group 1 and 2. So this group 1 and 2 coronavirus, we talk about the epidemiology. All 4 of the coronavirus have been reported from around the world. Most studies have focused on children, where this is more significant, although they infect adults. Infections are seen throughout the year, although more food is vector and strains with larger outbreaks every 2 to 4 years. This is about the group 1 to coronavirus. Pathogen that virus enters the respiratory tract, where they replicate the epithelial cells of the upper respiratory tract, spread to the lower respiratory tract. Spreading of virus from the respiratory tract to infection, and virus may continue spread for some time from the gastrointestinal tract after recovery. The clinical picture, as the previous speakers have already spoken about the previous virus, is that coronaviruses are important cause of the common cold 2 to 10%, second after 9%. In adults, the illness is related to common cold symptoms of rhinitis, sore throat, and sometimes coughing. In asthmatic patients and patients with chronic bronchitis and other chronic lung disease, the underlying illness may be worse. In infants, the infection can be more severe, causing tracheolaryngeal bronchitis, bronchitis, and about the diagnosis, due to mild and passing nature of the illness, diagnosis is very seldom required and usually limited to diagnosis in the recovery period or past infection with epidemiological test, because when it shows symptoms of then diagnosis. Few laboratory tests for coronavirus and few offer such testing as the diagnostic service. In more severe cases of infection in infants, many other more treatable causes are today. Easier access to rare diagnostic tests are serological tests. This is of limited. It is used for epidemiological purposes like taking the blood, serum from the blood. Then isolation method for cell culture, that is Verosis, and molecular methods are polymerase chain reaction (PCR), usually specialized. This group 1 and group 2 coronavirus have specific treatment, like actually it being studied and a lot of controversy with the actual treatment. There is no specific treatment for coronavirus. Treatment is symptomatic and as
Hello, Dr. Sunil.
heard from the
Sunil, sir. Hello.
Not specific treatment is given, but based on the symptoms developed by the patient or sometimes only
Hello, Sunil, sir.
psychological secure some treatment are. And as far as prevention is concerned, general hygiene and disinfection can prevent person to person spread.
Hello. Hello,
Yes?
Sunil, sir.
Yeah.
Sir, the participants in the chat box are complaining that you're not audible to them. So kindly, can you take out the headphones and while you're using your laptop, please? Sir, are you using your headphones right now?
I was. Is it audible or is it audible?
No, sir. No, sir. The participants are constantly complaining that you're not audible to them.
Is it audible now?
No, sir. Not yet.
Okay. Let's check it was.
So could you please take off your headphones and try it?
Yeah, I have taken out one.
But still, I think there's some problem with your audio.
Is it audible now?
Sir, can you raise the volume of your speaker? Hello, Sunil sir. Am I audible to you?
Is it fine now? Yeah.
Yeah, a bit okay right now.
Okay. So
Yes, sir. Please continue. Please continue. Sorry for the interruption.
Oh, it's okay. In fact, it's my issue also. Fine. So after the group 1 and group 2, the another is severe acute respiratory, that is SARS coronavirus. Talking about the epidemiology, that between November 2002 and July 2003, 8,096 SARS cases were reported to the WHO. The total number of deaths rose to 774, the case mortality of 9.6%. Since then, still about restricted with the outbreak occurred with a total of 11 cases. Since then, SARS has not occurred. So this particular SARS coronavirus entered the human population from an animal source. Several animals have been found to have closely related viruses. The epidemic began in Guangdong province in China, spread to Vietnam and Cuba, and then to other countries. Spread of the virus was via respiratory droplets, and therefore contact with surfaces was important. Since it was excreted via the intestinal tract, fecal and transfer was possible. And the incubation period of the SARS virus was 4 to 6 days, and patients were only infectious during the symptomatic period and most infectious during the second week of. Just you can see the effect of SARS coronavirus Mrna, which is having the still same structure. The pathogenesis that determines the pathogenesis of SARS was difficult based on autopsy findings caused by the effects of treatment, example ventilation and mutation. The virus originates in the lower respiratory tract, followed by an innate and specific immune response, and both viral vectors and immune responses play a role in pathogenesis. The first stage of the disease is associated with diffuse alveolar damage, macrophage infiltration, and type 2 lymphocyte stroke infection. The pulmonary infiltrate appears patchy on the chest X-ray. In the second stage, organized marker. The infection is not related to pulmonary system. The virus replicates in enterocytes, resulting in diarrhea and state in soup, as well as urine and possibly other body. The clinical picture is that first phase of disease consists of fever, which is more than 30 centigrade, with rigor, myalgia, sore throat, and gastrointestinal symptoms, with cough and shortness of breath after 3 to 7 days of symptoms. Hypoxia may develop and 10 to 20% require ventilation, related lymphopenia and neutropenia occur. Some patients even showed a biophysical course with apparent recovery followed by a of the physical condition. And mortality was highest in the elderly and lowest in the younger population. And coexisting illness worsened morbidity and mortality. This is about SARS. Diagnosis was that initially etiological agent has to be identified and was done rapidly through the methods that I showed you earlier. Serological it was based on the presence of antibiotic infection, then isolation through electron microscopy, cell culture, same thing. And another common thing here, that common thing is molecular testing is being done by the RT-PCR. And the treatment as supportive treatment such as ventilation was the centerpiece of the management of SARS patient, and specific treatment such as steroids became controversial regarding their effect, whether adverse or beneficial on SARS patient. Ribavirin was as is a broad spectrum drug and appeared to have some benefit, as did other agents, and that have an effect on the inflammatory response such as chloroquine and etc. And agents that interfere with coronavirus protease activities such as lopinavir, ritonavir combination for HIV treatment. And SARS specific was also tried. The degree to which any of them was successful as treatment still prevention as isolation of patients and quarantine of exposed people or disinfection and infection control precautions are the key to prevention of spread of SARS virus. Vaccine was also developed for academic measure, and since then epidemic is over and SARS is no longer threatened. So this was SARS coronavirus. Now we'll be talking about present for which is SARS-CoV-2. And as you know, many of you are aware of the symptoms. It will have fever, symptoms of shortness of risk factor is that the COVID have symptoms or mild, severe, or fatal. Kids may have this that rate that they have more immune supply. Current case fatality rate is 2% of those with level COVID-19. Risk factor for fever illness may include mortality or underlying chronic medical factors, comorbidity. You know this how being spread from the previous speakers, so I must talk on this. And treatment, as you know, there is no specific treatment currently available. Supportive management of complex include advanced organ support is indicated. Antiviral under investigation, and currently there are in the vaccine preparation there are 137 candidates under preclinical development and 23 in early clinical development according to the WHO. And ideal vaccine against SARS-CoV-2 would be effective after 1 or 2 vaccines. And as even in the phase 3 trials, most of time it is found to be negative. Now, the COVID pandemic few days, let me just go through this because it is told that in December 2019, the novel coronavirus was identified as the positive event of clusters of pneumonia in China. The virus was SARS-CoV-2 based on biological and taxonomic similarity to SARS-CoV, which caused an outbreak of SARS in 2002. WHO named as SARS-CoV-2 as coronavirus 2019, COVID-19. The first confirmed SARS-CoV-2 case was found in Wuhan, China. Subsequent workers and family clusters who had not visited Wuhan test positive for SARS-CoV-2, thus confirming that it was also transmitted from human. SARS-CoV-2 was spread to most countries and resulted in thousands of fatalities. So WHO that's why declared this pandemic on March 11, 2020. This rapid spread and is three contributed to the WHO, which has as the pandemic in the world. Now talking about few things where the structural studies or the role of bioinformatics or structural biology, just refer to this particular slide with the structure of SARS-CoV and SARS-CoV-2. You can see most of these having similar except theory. There are some of the non-structured proteins that can the structures which are available protein database. So genomic description of this SARS-CoV-2 is that the genomes of this SARS-CoV and SARS-CoV-2 are shown as white. You can see the open frame represented by green and colored boxes to indicate to have similar and different than respectively based on test conservation. As recently published in Lancet Journal 29 2020, genomic characterization and epidemiology of 2019 COVID virus implication for virus and vector binding. Next gen sequencing is sequenced by sampling from bronchoalveolar lavage fluid and cultural isolate from 9 patients, 8 of whom had get the Hunan. Then they did complete and partial sequences obtained with viral were connected using tanker sequencing to obtain full length with the terminal determined by rapid amplification of CDN and phylogenetic analysis of this COVID-19 genome, and those are the used to determine the evolutionary history of the virus and help its life geometry. Homology modeling was done to explore the likely receptor binding properties of the virus. The finding from this next-gen sequencing studies was that the 10 genome sequence of this COVID-19 obtained from 9 patients were extremely similar, exhibiting more than 99.98 sequencing length. But notably, this COVID-19 was closely related to 2 bad severe acute respiratory syndrome like coronavirus, that is bad cell COVID and bad SL4 ZXC21, which was selected in 2000 China, well, but were more distant from SARS-CoV and MERS-CoV, which was Asian. Phylogenetic analysis that this coronavirus 912 is the subgenus Sarbecovirus, the genus Beta coronavirus is related to long branch length to its closest relative, this back to back, and was genetically different from coronavirus SARS-CoV. But notably, after modeling was done, it was found that binary information that are structured to the coronavirus SARS despite amino acid at the key position. You can see some here that this particular SARS coronavirus is having more closeness to this bad coronavirus. However, it is distantly being related to the present coronavirus, that is COVID-2. Another study was the interpretation of this genomic characterization was these studies were significantly divergent from SARS-CoV-2 considered as infecting beta coronavirus. Although phylogenetic analysis suggests that that might be origin of this virus in Wuhan and the emergence of the virus. But importantly, structural analysis suggests that COVID-19 might be able to bind to the angiotensin converting enzyme, that is AC2 receptor, and the future evolution, adaptation, and spread of the virus current urgent illness. So here we are particularly more concerned about this thing only to study that what are the findings made by the researchers till now coronavirus. Studied a malignant relationship with the previously studied or occurred by coronavirus. So this study would show the phylogenetic analysis and it shows that phylogenetic analysis of RBC from various beta coronavirus are here. Beta coronavirus, the structural comparison of the receptor binding of SARS-CoV-2 and COVID and COVID-19 and SARS-CoV-2 also are seen here. So you can see here that how structurally based on the phylogenetic and homology model, how it is stored in this case. Another study on vector inside SARS coronavirus 2 receptor and antibody interaction unique to inside SARS-CoV-2 protein. This studies also showed, as previously somebody already told, that the structure of SARS-CoV-2 receptor binding shown here. You can see that thematic architecture of SARS-CoV-2 spike glycoprotein. Degree of protein surface concentration between primary SARS-CoV-2 and SARS-CoV-2 spike protein. The color shown is with green and magenta representing that these are not conserved and conserved region. The card figure also shows the structural alignment, and you can see at the C, these are the close and full confirmation of the SARS-CoV-2 with one of the RBD domains buried or exposed. Now, while seeing all studies based on structure, based on the earlier virus, what the area of bioinformatics has a whole combat coronavirus. So, as you know, that application of bioinformatics used in pathogen identification, detection of virus factors, resistome analysis, strain type. As you can see that the sequencing technology is also supported by bioinformatics, then phylogenetic and pathogenetic analysis help in the identification of some of the processes like cross-treatment, hemolytic. So these aspects were factor necessary to establish an infection and cause of all the other symptoms. Thus, this next sequencing holds considerable potential for pathogen identification, specific specimen sequencing method powered by your bioinformatics. This is very interesting figure to show that how this disease transmission in an outbreak is depicted in the layer form. You can see from the layer one that is the geography environment, where the disease is introduced to a certain geography, where some environmental factors may promote the transmission. The inverse lineage has selective advantage in detecting and transmitting a certain group of hosts, which is shown in population demography. In the clinical manifestation, you can see the actual spread of coronavirus. This shows the inverse association with distinctive presentation or severity, which could be altered by phenotype itself or the interaction. As some of the viewers were questioning about the herd and it developed in some, you can see that is reflected. Then the image exhibit a different viral phenotype such as vaccinating titular or duration, which is due to the common mutation of the virus. And in the genomic span, you can see that phylogenetically different from the genomic sequences serve as proxy of disease transmission. This is another very interesting figure. You can see that pangolin also harbors the SARS-CoV-2 related coronavirus. When the phylogenetic was studied in SARS-CoV-2 related viruses that it showed that SARS-CoV-2 related viruses based on all sites or the same site of the receptor binding cross-species transmission scenarios cross-species transmission scenarios pattern shows that pangolins might ultimately originate from cross-species transmission of bats. And you can see right here from the palette that pangolins serve as intermediate host and SARS-CoV are to spill over pangolins to human or multiple times from human to human. And the SARS-CoV and pangolin is originated independently through cross-species transmission from bats. And the black are cross-species transmission. That was the said that particular bat the virus might come from bat to pangolin to human. And this shows this palette. So our basis of studies the particular bioinformatics was that epidemiology investigation by the National Center for Disease Control and Prevention said that the outbreak was associated with seafood market. Studies were done on the patient who were on the market as we are saying. The next generation sequencing identified novel RNA viruses from the family coronavirus designed as human coronavirus. Phylogenetic analysis of the complete virus revealed that the HCV was closely related to a group of SARS-like coronavirus previously sampled from bat and that have a genomic. So based on all this kind of information gathered from the data survey and all kind of work going on the world, we could interpret that some of the tools which are already available and being studied on pathogens can be used to somehow combat this coronavirus scenario. So an important point tools to test pathogenicity of covered bacterial pathogen finder 1.1. These particular pathogen were used for the prediction of bacterial pathogenicity utilizing proteome or not. The bacterial depends on proteins known to what pathogen. These web servers utilize the selection of proteins created without annotated function or no involvement in pathogenicity. It can predict pathogenicity for all taxonomy of bacteria with 88.6 accuracy. So you can use this tool to get the raw data from the public data on coronavirus and find out the pathogenicity using this pathogen tool. A recent method for predicting pathogenicity is this paper bag called pathogenicity prediction for bacterial genome. It is based on machine learning and provided as the R package. So those who are familiar with R can do this. This predict also pathogen by means of training on a large number of established pathogen species in comparison to non-pathogen bacteria. It is suitable for evidence data with a very low genomic coverage. This particular paper bag is for this method for assessment of the potential of state of breed belonging to a genome. It helps the prediction of novel unknown bacteria. So based on your data collected on either samples from the local hospitals there or the public database, you can try paper bag to find pathogen. Similarly, the economic contigs of a pathogen produced by NGS techniques are annotated using these proprietary atomic annotation pipelines available here. Actually, pipelines are but the flow of tools being used for studying the next gene sequence data. So this PGAP is available at we have said you can search and follow. It can also be annotated in bacterial bioinformatics database and an analysis resource that is Patrick service for pathogenicity and factors. Virulence sequences and functions corresponding to different major bacterial virulence factors of a specific pathogen can also be collected from gene bank and validated using virulence factors of pathogen bacterial services like Victor virulence factor search program and Patrick BF2. So the need for accurate detection and understanding of resistant factors and mechanisms are highly demanding in antimicrobial resistance. As we when we go across this thing, unless we get some sort of lead, we don't actually apply the tool. So all these tools are developed based on its accuracy and kind of validation. So the genome predicts whatever we have studied on coronavirus. We are also studying on the same series. The genome predict can be investigated for the presence of antibiotic resistance loci using both this PGA and Patrick annotation services. Further, the presence of antibiotic resistance loci for the bacterial pathogen can then be investigated with specialized search tools and services named antibiotic resistance search or feature finder and this comprehensive antibiotic database specialty search and RES finder. This RES finder 2.1 identifies acquired antibiotic by antibiotic resistance and finds chromosomal mutations in total of partially sequence isolation of bacteria. So when we tried all these currently available bioinformatics tools and of the raw data in public domain, we found that with the help of various tools and data generated, we can actually supplement the medical based research on coronavirus. And we have seen some of the leads in our lab so when we studied on. We also have testing laboratories here. So some of the data we got from the hospital that also we are trying to study and we are getting of good results, I should say, because even here also there is a lot of variation and diversity in this virus which we are studying. And this particular this presentation I have covered that to find the antibiotic and the on the bacteria so as to study the preliminary physics of studying the virus. But there are other tools also available to find the phylogenetic relationship and the variation of viruses. We are also working on sequencing the microbiota of particular coronavirus, find that how it is diverse from other infect even so this was my slide last slide. Thank you.
Thank you so much, sir. Thank you so much. for providing us with the detailed information on the genetics of this SARS-CoV-2 virus. So the researchers among the audience of this webinar will be definitely intrigued by the new possibilities on working on the genomics of this virus. So in spite of the technical problem regarding the voice interruption, your slides were self-explanatory, and definitely the participants have extracted the useful information from that. Thank you, sir. Sir, here are some queries from the participant side, and I wanted to convey them. So one of the participant is asking about the mutation of the virus. Are there any specific receptors being identified which get changed in the virus?
We are also studying on the same issue actually when we are trying the molecular issue. It's been tried in the aspect because it is integrated with the heart and blood vessel. So this particular we are trying, but we are able to find some of the mutations because right now we are only working on some of the publicly available data. So it is difficult to say, but definitely there is mutation as we are studying on the other aspect of viruses to India itself. We find there is there are mutations, but it's not not fair to say that on what of course there are some of the variations found, but the exact cause is not. But we can yes.
Okay, sir. So one more question related to this mutation only. That do you think that coronavirus strain affecting the Indian people are different from the strain found in other countries, as the symptoms are also somewhat different?
As previous speaker also answered, like the lifestyle, they say that depend on the lifestyle also some of the some of the patients are cured or much infected or not much casualties are there. So there are like people bring various things, but scientifically not true that whether the lifestyle is the thing like your food habits or genetically. Because as you know that we are working in the northeastern part of India, earlier there was no spread of this particular virus. So there were some theories that make this part had separately exposed, so there will be some resistance. But it was contrary to it because the origin was also from South Asian countries. So, but now it is spreaded here also. So it is difficult to say on that aspect unless you study there. And let me tell you that we are also very beginner in looking at on this particular virus, but definitely the scope is number one research. But particularly research in Corona. So of course, very much are there, but can't be.
So one more question. So how far the homology modeling could be helpful for treating this disease, as mutation rates are variably high in this virus? So how the homology modeling could be helpful for
Homology,
making the treatment of this virus?
yes. As we are also modeling for a particular suppose structure from the public domain databases, and surely it may vary also. Suppose if you are working on just particular protein, uh, modeling on one protein suppose, so it will show different. That is the reason it is not now sure that whether the vaccine on basis of the clinical studies or the structural studies will be actually effective. So we are trying to study more number of samples, and that we only should be able to find out. But definitely that aspect also we are finding that though the results are very clear in one particular set of models, but then it may not be same with the other. So we can't actually say on that. But we
Okay,
have
so
to
thank
keep
you
on
so
trying
much.
because
Thank you so much, sir, for such an informative session, and we are really benefited from your talk. I now request Ujjwala ma'am to be online and deliver the vote of thanks. Ujjwala ma'am.
A warm and graceful good noon to all. The most valuable, honorable resource persons, Chief Patron Dr. Manager Bishop, Patron and Administrator, Principal Dr. M.G. Royman, the worthy teaching and non-teaching staff of St. Thomas College, students, and all the participants. It's my proud privilege to propose a vote of thanks and acknowledge the contribution for those who work hard to make this event successful. I extend my most sincere thanks to Almighty God for making today's event successful. I would like to thank our chief patron, Dr. Manager Bishop, to help to organize this international webinar. I am extremely thankful to our administrator for taking effort to organize this event. A special mention to our respected principal, Dr. M.G. Rahman, for being catalyst and stimulated us to do our best. I extend a sincere thanks uh, to our all respected resource person who spared time for the busiest schedule to grace this international webinar. Today we had an opportunity to hear their thoughts, and this will be surely going to encourage us in our future events. I would also like to thank the people who work hard behind the scene to make this event happen. I extend thanks to Mr. Michael for his technical assistance. And last but not the least, I am, on behalf of St. Thomas College fraternity, thankful to all the participants and students for making this event as a grand success. Thank you. Namaste.